speaker is Roichman, and she will actually present the title since she changed the title from the one that we have. So.
Okay. Hi. I'd like to thank the organizers for inviting me so very kindly and for a lovely conference that has been actually a lot of fun. I'd like to also apologize for changing completely my talk. And the reason is that I'm naturally mostly excited about what I do at the moment, and this is what we just finished doing at the moment. As we're speaking, we're writing the article. So what I would like to tell you about is life in a gel, or the more technical title is the viscoelastic response of complex fluids in the intermediate regime. What I am actually going to talk about is entangled F-actin networks as complex fluids. So this is a gel of a semi-flexible polymer. This is in vitro, so we polymerize this in a tube in the lab in the presence of tracer particles, and we follow them. But when we were working on this, we realized that we are actually seeing results that are more general than that. So the outline of the talk will start with something that is completely irrelevant and has nothing to do with what I'm going to talk about, but it is what we're doing in the lab, and I hope it's exciting not only for me. Then we will continue to talk a little bit of things that are somewhat relevant to the project, and this is describing what is microrheology and what are complex fluids. Then I'll finally get to the point and tell you what is so exciting about what we do and what we found out. And finally, if time permits, and you will have to tell me, I will tell you a little bit about what I originally set out to tell you. So let's start by some general things that we do in the lab. So originally, what I'm doing is optical trapping and optical imaging. And what you're seeing here are particles, silica particles that are being trapped, or nanowires that are being trapped and manipulated. We have recently built a system which combines optical trapping and confocal imaging. So you can trap, this is a 3D rendering of something that actually we did trap, two different types of materials to create photonic structures. You can trap, because you can measure things in 3D, you can make sure that where you place something in space, it really goes to where you want it. So you can make a perfect cube. You can also trap arrays of cells and then characterize them. So what I'm going to show you here, this cell, which is this cell, is trapped by 2 point traps. And the main question is it still happy? Is it still alive? And one way to see if someone is happy if it reproduces. So what you're going to see here is this is the nucleus, and the green thing is the cytoplasma. And you're also seeing here a sequence of imaging from the images from the same movie. And what we're going to see is how this nucleus is transferred from the mother cell to the daughter cell. And here you're going to see us measuring from fluorescence the amount of DNA material in the nucleus. This is of the mother, and this is of the daughter. So let me show you the movie. So you can actually see them splitting, and so this is really nice because it means that we can localize 10, 20 cells and characterize their behavior in real time. Different kinds of imaging that we're doing is holographic imaging. This is similar to what Nati does, only this is inline holography, which means that I'm using the same beam for imaging and reference, and so the analysis is slightly different. What you're seeing here is raw data of particles flowing in a flow cell. And this is, from this data, we can extract the 3D position of the particles in real time as fast as the camera goes. So what you could do is you can follow a particle which is floating above. This is a glass wall, and the particle is floating here above. You can calculate the distribution of heights that the particle is experiencing, and from this extract the potential that the particle is sitting in. So the electrostatic repulsion from the glass and the gravitational forces. You can also use it for tracking. These are two attached particles. One is 2 micron in the cross-section, and one is 1 micron cross-section. They're stuck together, and they're diffusing in 3D. And I can get their 3D trajectory, and I can calculate from this image the distance between these two particles. And it's exactly 1.5. It should be exactly 1.5. It's very close to that with nanometer resolution. So I'm also in the Nano Center. Other things that we're doing is particle tracking. So many of you have done that. You look at, say, you take an image, you look at the particle, you find its coordinate, you take a movie, and you connect all these coordinates, and you can track the trajectory of the particles. And I'm going to use this technique for the entire talk today. But there's another way to characterize a particle, and it's called particle image velocimetry, which what you do here is you divide your image. This is a fluid containing colloids, and it's being sucked into a wafer with pillars. So capillary forces are pushing the liquid in. And I cannot follow all the particles here in because I don't really, they come in and out of focus very fast. But I can divide this into pixels and look at the correlation from one image to the other of this pixel. And from this I can extract the flow field. And so what you're seeing here is the flow field of the water imbibing into the surface. And you can see that the capillary forces are causing higher velocity in areas which are more compressed and more smaller. So this was for the exciting talk, and now for the actual talk. So what is microrheology and what are complex fluids? In microrheology, what you do is you do the following. You track particles, in this case in water, and from their mean square displacement, which is plotted here, you can extract, using the generalized Stokes-Einstein equation, the elastic moduli of the material. So you can get elastic properties of a microscopic sample just by following a single particle. There is another, and also this is water, so you have only a loss modulus. Another technique to do this is to look at two particles and to look at the correlated motion of these two particles. If you look at the correlated motion of the two particles, you can also get something which is similar to a mean square displacement. You can also transfer that using the generalized Einstein-Stokes relation to the elastic moduli. And this is a way to characterize it. As you see here, the 1 point, it's called 1 point and 2 point. 1 point and 2 point microrheology schemes give you exactly the same mean square displacement and hence the same elastic properties. When you go to actin networks and you do the same, so this is the green is the 1 point microrheology and the red is the 2 point microrheologies, they don't agree. And that was a big issue because the whole idea of microrheology was to be able to measure elastic properties, and if these two don't agree, then which one is right? Okay, so this is the 2 point microrheology. Another thing I want to make sure you notice is that they are very different in moduli. So what the 1 point microrheology measures is something a lot softer than what the 2 point microrheology is measuring in an actin network. Okay, so what are complex fluids? Complex fluids are all around us. It's from ice creams, creams, paste, to things that I like more, like granular piles or M&M packings, or things that I can actually see in the lab, which is milk or a colloidal suspension or an actin network with myosin inside. These are all complex fluid, and what makes them so nice is that they're all individuals. By this I mean they're all made of mesoscopic objects, and they flow. So if I give them a shear stress, they will respond to it. If it's strong enough, they will flow. But they also maintain some sort of integrity, so there's something in the middle, and they're viscoelastic. This is what we mean when we say they're viscoelastic. So now to the point, what I'm going to tell you about is the viscoelasticity of complex fluids, and specifically in F-actin networks. All this work was done by Adar, and she actually figured out most of it. I don't know that I could do anything without her. And this is in collaboration with Chaim. Okay, so back to complex fluids, and I want to now tell you how we understand these systems from very, very general considerations. So here is a network, but this could be also a colloidal suspension. And as I said, all of these things have mesoscopic building blocks. So there's a length scale in the system. For instance, the mesh size of the network, which is the average distance between filaments of the network. And in our case, with microrheology, we also have the size of the particle, which gives you another length scale. So you know that the elastic response will have to depend on length scale and have to have some sort of time scale in it. It cannot be just the simple one-point microrheology. Okay, so now I want to ask you the following question. Say I have a three-dimensional material, no boundaries, and I put a point force pushing this particle slightly to the right. What will be the response of the material? So first of all, there's my momentum. Consider the momentum that I created. It's a monopole momentum, and then it has to travel in the system. And just by considering momentum conservation in 3 dimensions, I know that this thing has to decay like 1 over r. It's a monopole. There will be other decays, like 1 over r third to the power third or so, but they are negligible. And how does this flow field look? So if I push the particle a little bit here, you can see that all the water is going to follow, but in a peculiar pattern. There's another thing that I do when I move these spheres. I'm moving mass. I'm creating a mass dipole. And if I do the same considerations for a mass dipole, then it has to conserve mass. Mass is not going anywhere. So just from consideration of mass conservation, this has to decay like 1 to the power of 3. And this is how it looks like. It's a dipole flow field. And when you combine both of them, you can see that you don't have to go very far to go back to flow that looks a lot like 1 over r. So at large distances, 1 over r is what wins.
Mass dipole is higher or lower than the density?
Yes. And basically what I've told you now is why microrheology is so successful. All I have to do for any material, provided momentum is conserved, if I look far enough, the decay has to be like 1 over r, and I can extract from this elastic moduli. So this is the strength of microrheology. I can do it on actin networks. I can do it in cells. I can do it, well, maybe there the momentum is not conserved. I can do it on colloidal suspensions. I'm free to do it wherever I want, and this is great. You can ask me, when I'm saying that the response, the momentum response is everything I see, how far do I need to go in order to see the mass? Is it interesting, this mass decay? And the answer is, well, I have a length scale, so probably the crossover is somewhere around this length scale. This length scale in actin networks is 300 nanometers. Not very important. I can forget about this. And here comes the but. When you look at the mass dipole term, it has to do with the local environment of the particle. Because what I'm doing is I'm moving mass near the particle. When I am looking at the momentum monopole, I'm looking at something that travels throughout the system. And usually, if I want to look at correlations at far distances, I have to do 2-point microrheology, so I'm looking at 2 particles that are very far apart. They don't care what the local environment is. All they care is the bulk. So this changes the ratio between, or the importance between the local mass dipole effect and the bulk momentum decay. And so it defines a crossover. So the length at which I can forget about the mass dipole and go back to looking at elasticity depends on the square root. It's from this, just by turning this to 0. The square root of the bulk viscosity versus the local viscosity. In water, there is no difference. That's why 1-point microrheology and 2-point microrheology gave me exactly the same. So in the case that they are equal, I will get a flow field that is very similar to 1 over r. But in the case that the local environment is a lot softer, I will get this effect much further apart. And the question, of course, is how far apart is it? So what have I said so far? I said that microrheology works so well because it has to do with conservation laws, and it's very general. I said that the intermediate response, or the mass response, is also something that is very general, also material independent, mostly, and that the crossover between the regimes has to go like the ratio of the viscosities. This means that for any complex fluid, I should be able to see that has this effect, and this is usually right. I will see the intermediate response at larger distances than I would have expected. So it's larger than the mesh size, for instance, in Actin. So all I have to show you right now to prove that this is something interesting is that, first of all, that this effect is actually relevant for some real system, and to show you that this is experimentally accessible, that I can actually measure something like this in length scales that are of interest. So what I'm going to do is I'm now going to concentrate on an example of Actin networks. And theoretically, Actin networks have been shown to behave or conform or be described well by the two-fluid model. What is the two-fluid model? The two-fluid model assumes you have two fluids. One of them is an incompressible fluid, which is water, and the other one is a flexible or semi-flexible polymer, which is the actin. And what goes between them, they are coupled through the friction forces and the relative velocity between the polymer and the liquid. And this is all you need to know for a two-fluid model. It could be any polymer network. And if you ask Chaim to do the calculation, he will. He looks at it and says, this is not so easy. I think it will take me an afternoon. Anyway, he goes there and he does this, and he shows indeed that the crossover has to be depend on the ratio of the square root of the ratio of the bulk viscosity to the local viscosity, and also that it has to depend on some function that scales like the dynamic correlation length of your system to the size of your particle. And if you do this is actually very general, but if you do this for the two-fluid model, you know this function. This function has to be x squared plus x plus third. Why? Because this is how it comes out in the calculation. I just want to let you notice one point. This correlation length, dynamic correlation length, that can be measured usually only by light scattering, is not the mesh size. It is related, according to Dejean, by some factor, which is somewhere between 1.2 to 1.3. But it's not the same. And again, just to show you the local environment. So what do I mean by local environment? Here is my tracer particle. It's been there when I polymerized the actin. So the actin is around. It's a semi-flexible polymer, and it cannot penetrate and touch the sphere. The sphere doesn't adhere to the sphere. So there's some volume in where the density of actin is lower near the sphere. So this is a lot softer than what goes in the bulk. And this is the reason why we have this difference between local and bulk viscosity. So now, if you look at the one-point microrheology, which follows basically the local environment, it's a lot softer than the two-point microrheology, which gives you the stiffer response, the long-range response. And this has been compared to bulk rheological measurements, and we all know, or at least in the field, that the two-point microrheology gives you the real deal. This is the bulk viscosity. So, this is somewhat unexpected. Okay. Sorry about that. So what you go now and do is you calculate this a two-point correlation motion of the particles as a function of distance. And everyone has been using, up till now, only this range. Nobody has looked at that range for two reasons. One, you need to do a very good experiment with a lot of statistics, and only Adal can do that. And the other thing is that nobody expected anything interesting to be here. So people concentrated on the area where they could extract 1 to the power of r, and then this is what they used to get the elastic modulus. But Adal came to me with this plot and said, listen, something is very, very different than what you expected. And we tried to explain this in various ways, and in the end, Chaim came to our rescue. Another thing that happens if this is really the mass... So up till now, I haven't proven that this is the mass dipole effect. It's just an effect. And to get a power here in a logarithmic plot with very few points... So I haven't proven anything yet, but if this is the mass dipole effect, then I expect the correlation in the transverse direction to be negative at the same area. And so we do see that, although this is very, very noisy and hard to extract. Okay, so this is just repeating the fact that it could be the crossover, and if it is, it's about 3 microns. So this is a very large distance. I have a mesh size of 300 nanometers, and I'm seeing the intermediate response up to 3 microns. So can we extract from experimental data the ratio between the bulk and the local viscosity? Because this is what is interesting. So Adam came up with a trick to do that. And so we're saying that there is some prefactor to the 1 over r cubed, one prefactor to the 1 over r. This prefactor can be correlated or described in terms of the 1-point microrheology. This one can be described in terms of the 2-point microrheology, and the ratio can be shown to be exactly, or almost exactly, the ratio of the viscosity. So now, let's look at the crossover behavior and see if it agrees with this thing. So originally, we had plotted the crossover as a function of lag time, because when you do mean square displacement, this is what you do. You plot it as a function of lag time, and we couldn't make sense of this plot. But if you plot it according to the square root of h, which I remind you is the square root of the ratio of the viscosities, you get a straight line. So this is great, but from the general considerations that I told you before, this is true, should be true for any complex fluid. It doesn't mean that the two-fluid model is actually predicting what we're doing. So what you want to do is to see how it depends on psi and A. And then you can say what goes on. So this is the theory. This, if you square the equation and you replace the ratio of viscosity with the measured ratio of viscosity, this is how things should behave. And now you do tons of experiments in different mesh sizes and two different tracer particle sizes, and you see that they all behave linearly as you would expect. And if you put them all on a single scaling curve, so r squared divided by h and a squared, as psi, and here we put the mesh size, psi a over a, then this should all fall on a line with a function that goes like x squared plus x plus a third. And at least for me, when I saw this, I couldn't believe it. This is almost too good to be true. We could extract from this the fitting parameter, which is the ratio between the dynamic correlation length, psi d, to the mesh size. And it came out to be 1.25. Which means that we can measure with this technique the correlation length in the system, which was previously done only with dynamic scattering, light scattering technique. So this is a new way to measure the correlation length in the system. And also gives you, so now I completely believe this entire analysis. We have a two-fluid model for the actin. We can characterize the elastic response from the intermediate regime to the long-range regime. We can tell you at what length scale this is important. And if you look here, we're talking about crossover lengths from 2 microns to 6 microns. So just to sort of summarize what we have up till here, we've now provided a generalized framework to look at microrheology, which is also system independent. And it connects the entire range from intermediate to long length scales. We can measure dynamic correlation lengths. This should be true for any complex fluids which the local viscosity is smaller than the bulk viscosity. And we get it at the crossover lengths to be more sometimes than 10 times the length scale in the system. And I would just like to compare this scale, 2 to 6 microns, to cells. So this should really be important in systems of interest. And just a final thing to note, that sometimes when momentum is not conserved, this intermediate response, this subdominant response, is all you have. So it's really, really important. Do I have time? Okay, so in this case, I'm going to show you just a teaser. So when you do microrheology, the first thing you have to do is you take your trajectory and you look at how much, at a certain time lag, the system is propagated on average, and you plot the histogram of this step size. And if it's a diffusive process or a thermal process, you know that it has to be a Gaussian. And then you take all of this, you take the variance of this curve, and you plot the mean square displacement. And this is for F-entangled actin networks with no myosin. Myosin is the motor that knows how to attach to two actin networks, actin filaments, and slide the one against the other. So this is, in a way, it does use ATP, the system. It polymerizes and depolymerizes, but it's mainly did. Now you can add some motors into the system. And you have to be very careful how you do it and how many you put. But if you, this number here, A to M, what it means is how many actin monomers I have in relation to myosin monomers. So the larger it is, the less motors I have in. So if I change it from 600 to 120, and I look at the histograms, they're slightly...
Exactly, histogram for tracking particles.
No, so this is, I'm tracking particles, and I'm looking at the histogram of the displacement after a certain lag time. And so they become narrower now, and you can see that the mean square displacement becomes smaller. Now, would you have expected this? I actually expected the exact opposite, because if you put something that moves things, then everything should diffuse very aggressively, not become confined. So this is one puzzle. And then you say, okay, maybe it's not enough motors, let's put some more. And it becomes a lot, oh, this is a bad choice of color, a lot more narrow and a lot stiffer. And then you add more motors, and now you're seeing two things. First of all, it's really stiff, but also you have a new feature here. So I cannot account for this new feature yet, but what I can tell you is why everything becomes stiffer. And the reason is that when you put the motors in, they move, they slide everything against each other, but they also oppose each other. And what happens is that your system, instead of being some semi-flexible polymer, is now a stretched polymer, a pre-stressed polymer. And this thing is actually very important for cells, because they wouldn't be able to transfer mechanical signals if everything wasn't pre-stressed. So this is, when you stop to think about it, this makes a lot of sense. And so with this, I'd like to mention Chaim again, which is a great collaborator, Anne from Beer Sheva, who harvests the actin for us. Adar, that did everything. David was the holographic imaging guy. Maya and Dror did the confocal microscopy and funding agent. And thank you.
It's time for a couple of questions.
Can you change effectively the local environment by modifying the particles, by adding some way for them to like more or dislike more the filaments, the fibrils? If you do it in a control way, can you see that the 2 point and the 1 point are actually changing?
If you don't coat them with BSA or some other PEG or something like that, then they attach to the actin network. And then what you're going to see is only the actin, or mainly the actin network. And this is a known artifact of people who don't know how to do microrheology. I mean, this is known forever.
And then the 2 point and the 1 point microrheology looks the same?
In 2 point, you always have more information, because you always have a length scale dependence, because it's a correlation depending on length. In 1 point, you're looking only on the entire system behavior on one, so I'm not sure if it's going to be exactly the same.
Continuing on the same, I just now that Simon dropped, although we've talked about it a lot, that most of the literature work characterizing these kind of materials use the 1 point, to my knowledge.
The older ones.
Okay. I only know
there is a reason for this. So if you need n data points to get a good 1 point microrheology, you need n square data points to get 2 point microrheology, which means that you need about 1 million data points. Which means that you need to measure, to make either a lot of movies or to measure everything for a long time with as many particles as you can. And again, you need a DAO for that. It's not so simple.
And is there a rule of thumb that defines the size of the particles that you have to trap in relation to the properties of the material?
Well, first of all, if your material, what sets the length scale is the length scale of your material. So the mesh size of the actin will tell you what kind of response you're going to have. If your particles are smaller than the mesh size grammatically, then you won't see many things because they're just going to go between. So you need them to be bigger than the mesh size. So we have mesh size of about 300 nanometers and particles of 1 micron. And this is already plenty. We also did with half micron particles, and we did see, we still saw the response. So these were the two sizes we used.